to the review today. So I'm just going to be going over a basic review of each of these worksheets. I know that there's been quite a few questions, and just due to the chaotic nature of the week, I didn't get a chance to review with you guys. So what we're going to do is go through a few examples for each of the different questions, and then hopefully that'll give you guys the ability to complete the rest on your own. Do your best. If you do have any questions along the way, you can ask me via email. I'll do my best to be checking it. Also, yeah, that's kind of it. So let's just go ahead and get started here. So I kind of went over both of these tables beforehand, but I'll do an example for the top row of each of them just to make sure we're comfortable. So for this first table here, what you want to do is tell me, based off of the antigen that's present on the cell, what your blood type is, and then what antibodies would react to it. So for example, if I have a red blood cell that has an A antigen present on the surface of the cell, then my blood type is type A. And then the antibody that reacts to this antigen is called anti-A. So that is the way to complete this. So based off of the B antigen being present on your cell, what would then the blood type be, and then what antibody would react to it? Same with AB, and then neither means that I don't have A or B proteins on my cell, which means that I have a naked cell. And that is then the blood type that you can find the answer to either here in the packet that has a bunch of information in it as well for you, or you can find it in the slides. Now for this next table here, the expected agglutination responses is essentially you telling me whether or not you would expect a reaction between the antibodies and the blood type. So for example, if I have blood type A positive, then I would expect a reaction between the antibodies for the A protein because it's on my cell. That's why I have type A blood. So I would say yes. Now with the B antibodies, they would react to the B protein. I don't have the B protein on my cells because it's not a part of my blood type. I'm A positive, which means I have A proteins, no B proteins, so I'll say there's not going to be a reaction. And then finally, for the Rh factor, remember this tells me if I'm positive or negative. If I do have the Rh factor, then I'm positive, and if I don't, then I'm negative. So if I have A positive blood, that means that I do have an Rh factor present on my cell, which means that I would expect a reaction with those antibodies. Remember, those antibodies are looking for those proteins. If they find it, then there would be a reaction. If they don't, then there's not going to be a reaction, which is why I do not see a reaction with the B antibodies because I don't have a B protein on my cells. And let's do this example down here for O negative. So O is naked. It doesn't have either protein on it, so I would not expect a reaction with the A antibodies because there's no A proteins to react to, nor would I expect a reaction with the B antibodies because, once again, there's no B proteins for it to react to, and because I have a negative blood type here, that also means I do not have any Rh proteins, so I would not expect a reaction with the with the Rh antibodies. So that's how you're going to complete this chart here. This question is just a matter of posting pictures of your sample plates. You can either take them if you did the lab in class, or you can use the lab video and take screenshots there. And then when you're completing the reaction table, same thing as up here. You're just telling me whether or not you saw a reaction with the three different serums. So serum A has A antibodies, serum B has B antibodies, and serum D has Rh antibodies. So for example, your sample 1 might have looked different than mine, but I'll just do mine as the example. I did see a agglutination response. I saw a clumping in my blood when I exposed my sample 1 to an A serum, so that means that there are A proteins present. I did not see the agglutination response with the B serum, so there's no B proteins present, but I did see the agglutination response with the Rh antibodies, so that means I do have the Rh proteins, which means that if I have A proteins and Rh proteins on my cell, then I am going to be A positive. And then that's, of course, how you'll complete the rest of these charts based off of your different sample plates. So these pages here, and let me scroll down here. Hold on, I need to change my tool. Here we go. These pages here and here have a lot of information that will then help you as you're answering the following questions. So highly recommend reading through. I know it can seem like a lot of text, but you already know a lot of it. It's more so just helping you provide context for the information that we learned in class and what we saw happen on our plate. So this question here, the presence or lack of different blood proteins determines whether or not you might be a universal donor or recipient. Explain why O negative is a universal donor and AB is a universal recipient using the following terms: agglutination response, antigen, and antibody. So O negative is awesome because it doesn't have any proteins on the surface of the cell, which means that there's nothing for my antibodies to react to. If there's no A proteins, then my A antibodies won't react. If there's no B proteins, then my B antibodies won't react. And if there's no Rh factor, then my Rh antibodies won't react to it. So that means that no matter what blood type I have, I can get O negative because it kind of flies under the radar of my immune system. There's nothing on the surface of the cell for it to react to. AB positive is a universal recipient because their body isn't going to recognize any other blood type as foreign simply because of the fact that the way that it determines whether or not there's a foreign blood type is by finding antigens that it's never seen before. If I have AB positive, I have literally every single type of antigen present in my body already, so it's not like it's going to show up as anything new. It won't seem foreign because it matches what I already have. So if you were to give me A positive as a donation and I'm AB positive, well, I already have A antigens in my system and I already have positive antigens in my system, so I wouldn't react. I wouldn't see it as being foreign. And then these last questions here, these three, are more so based off of what you think. This one down here is information that you can get from the slides. And then finally, doing these Punnett squares here, the main thing you need to remember, which I have right here for you guys, is which ones are positive, or rather, which ones are dominant and which ones are recessive. So if my A and B are both dominant, which means that you can have both of them expressed, and O is recessive, then if I have two A's together, then I have type A blood. If I have two B's together, I have type B blood. If I have AB, then I have type AB blood. If I have AO, type A, BO, type B, two O's will give me type O blood. So you can only have type O blood if you have two O's together because they're recessive. And same with the positive or negative. Positive is dominant to negative. So if you have a positive allele present, then your blood type is going to be positive. You can only have a negative blood type if you have two negative alleles. For this one down here, you're paying attention to both the letter and the Rh protein. So it follows the same exact rules. A's and B's are both dominant. You can inherit both of them together. So if you have, for example, let's look here. So if I were to bring my A positive across and my B positive across, then this is what my offspring's genotype would be. So that would be AB positive. If I were to bring this one down here, then this would be O. So that would be B positive. Down here, let's do this one. Then it would be, this would be A positive, because remember, positive is dominant to negative, A is dominant to O, B is dominant to O. A and B can both be inherited together. That's what's known as codominance, which we're going to learn about next week. So that is basically what you'll need to know to complete this packet here. If you guys, once again, have any other questions, you can send me an email. I'll keep my eye out for those, but this should help give you all the information that you'll need to know. So let's get out of this packet here, and then we'll go to this one instead. Whoop. So these first questions here, these are application questions. You learned all of this information either before spring break or from the slides today. It might not be exactly the way presented in the slides, but it's just applying that information to then answer questions that look a little different. So for example, what does it mean for an individual to be a carrier of a trait? So this goes into your genotype. If I have heterozygous genotype, for example, for eye color, I have one allele for brown eyes, which is dominant, and then one allele for blue eyes, which is recessive. I then am a carrier for the recessive trait. It's not shown in my phenotype because I have brown eyes, but I do carry the allele, so I can pass it on to another generation. If, for example, my partner was homozygous recessive and they had two recessive alleles for blue eyes, knowing that we get one from mom, one from dad, if I pass on my recessive allele and then they inherit the other recessive allele from dad, then they'll have blue eyes. Even though I have brown eyes, I carry the recessive allele, which means I can pass it on to my offspring, okay? And then these two questions down here, I know it's a little confusing. It says heterozygous, but then both of the letters are uppercase. I was having a problem with the font. I thought I fixed it, but clearly I didn't. Pay attention to the word describing the genotype and not the genotype itself, because this is heterozygous, which means it technically should be capital A, lowercase a. Whoa, it's not letting me do my little typey thing. Here we go. Capital A, lowercase a, and then same down here, it should be capital B, lowercase b, because they're both heterozygous. And then you can basically use your knowledge of genotypes, dominant and recessive, to answer the rest of these questions. Finally, this question here, when it comes to individuals with recessive genotypes, the reason why we love seeing recessive phenotypes on our pedigree charts is because we know what a recessive genotype is going to be. You can only have the recessive phenotype if you have two recessive alleles in your genotype. When it comes to a dominant phenotype, that's a little bit harder to predict because they might be homozygous dominant with two dominant alleles, or they might be heterozygous with one dominant and one recessive. Either way, they're going to show the dominant trait. So it's harder for us to know what their genotype is. Recessive individuals are always going to have two recessive alleles. Otherwise, they wouldn't have the recessive phenotype. So that's why they're really helpful. Those are like the things that we know for sure that we can then kind of plug that in and use that information to then fill out the rest of the table by looking at who's passing on which alleles. And that'll make more sense on this next page here. So these are Punnett squares. You guys know how to do Punnett squares, hopefully. If you don't, basically you'll just put in your different alleles. So once again, you want to remember that you want to pay attention to the description and not the actual genotype that's typed, because I had a weird problem with the text. So if I have a homozygous dominant and I have a homozygous recessive, then it would be lowercase c for the recessive and uppercase C for the dominant. And then it's just a matter of bringing them down and across. So this would be like this. And then you'll interpret that to let me know how many offspring have the dominant phenotype. I'll give you a hint. You want to look to see how many offspring have a dominant allele present. It's all four of them. And then this one down here is they're both heterozygous, DD, which would be, once again, heterozygous, so it's one dominant and one recessive allele. So then here, when we take that to our pedigree chart, remember, recessive alleles are, or rather recessive phenotypes are amazing when it comes to completing a pedigree chart, because that tells us for sure the phenotype and the genotype of those individuals. So if I say that the affected trait is to have pink flowers and the unaffected is to have purple flowers, then I can go right away and say anything that's pink is going to have two lowercase p's because they're recessive, which means they have two recessive alleles. And so I can fill all of those in just based off of the color of the square. And then using this information, I can then determine what alleles are passed on by the parents, and then that'll also help me understand what alleles might be for the ones that I don't know for sure, if that makes sense. So let's work through this one. This is where you'll want to do some Punnett squares along the way, because it'll tell you how those offspring will inherit the alleles from their parents. So for example, if I have two individuals, one is recessive and one is homozygous dominant, I know all of the offspring are going to be heterozygous because I just did a Punnett square like that up here. So I can say that all of the offspring are heterozygous. Sorry, this text color is not the best. Let me make it white. There we go. And there we go. And then this one. So I'm able to fill in the offspring, and then I have all of my recessive individuals also filled in. I have 3 more squares that I have to complete here. And that's where I'll use the information from the offspring to try and figure out then the genotype of the parent. So if, for example, this parent was homozygous dominant, there's no way that I would have a recessive offspring, because if I have to inherit one recessive allele from mom and one recessive allele from dad in order to be recessive myself, if dad doesn't have a recessive allele, that means that I won't exist, which means that we know right off the bat for a fact that this individual is in fact homozygous dominant. So we'll cross off or not homozygous dominant, they're heterozygous. We can cross off rather the homozygous dominant. So that one's definitely going to be homozygous, or words, heterozygous dominant. You can tell it's the end of the week, huh? And then based off of that, if we have 2 heterozygous parents, then there's a chance that we could have a homozygous dominant offspring, or we could have a heterozygous offspring. This is one of those instances where we don't know for sure, and that's okay. We can just say or. For this one, though, if we have, well, let me explain this a little bit further. So if I could have either homozygous dominant or heterozygous, homozygous dominant means I'm getting one dominant allele from both parents, and then heterozygous dominant means I'm getting one dominant allele and one recessive allele from both parents. Once again, completing a Punnett square to kind of map this out will help a lot. And that's where this Punnett square is kind of this exact match, so that might help. And then finally for this one down here, this one's pretty easy. If we know that one parent is heterozygous and one parent is homozygous recessive, well, we can't inherit 2 uppercase P's because there's only one out of the 4 different alleles, which means that we know that since this individual has a dominant trait, they need at least one dominant allele. Since they can only get one dominant allele, that means that the second allele is recessive. Hopefully this helps make sense as to how to complete this pedigree table here. And then you can use that information going onto the final page to create the tree for your family. So when you're completing this pedigree tree, you want to follow 3 generations: grandparents, parents, and then yourself if you have any siblings. I know everybody's family looks a little different, so if you don't have any siblings but you have cousins, maybe you can incorporate them. If you don't have any aunts or uncles, that's okay. If you only have one parent, maybe they have an aunt or maybe they have a sibling that you can choose, so maybe you have an aunt or an uncle from there. And then if you have 2 grandparents, awesome. If you have 1 grandparent, then you maybe can ask if anybody remembers the other grandparent. So do your best to fill in 3 generations, but I also know that there might be some gaps, and that's okay, because everybody's family is different. As I had mentioned, though, you don't want to use a trait that everyone in the family has, because then it's going to be a lot harder to track through the family. What you want to choose is a trait that maybe some people have and some don't. So for example, if I have a brother who has blue eyes, but I have brown eyes, and my mom has blue eyes, but my dad has brown eyes, then that would probably be a really good trait for me to follow, because then I can see which alleles go where. But if we all had brown eyes, well, I'll have a really hard time figuring out how to follow those alleles through our family, because once again, having a recessive trait present on your pedigree tree is going to help a lot when it comes to then completing all of the different members of the family and predicting their genotypes. So those are the 2 worksheets. Hopefully this review helps you in completing those worksheets. Once again, do your best. If you do have questions, write them on the worksheet so I know what we need to spend more time on next week. Otherwise, I am excited to see you guys. I hope you have a great weekend, and I'll see you next week. Thank you.